Hello everyone, welcome back to my channel. In this playlist, we are going to explain about hematology, what are the formed elements of blood, how they are formed, and what are the diseases of blood. So let's get started. So what does hematology mean? Hematology consists of two words. Hema, which means blood, and logos, which means science. These are Greek words. So at the end, we will have the hematology word, which means the study of blood. In fact, our body constitutes 5 to 6 liters of total blood volume, or 7 to 8% of body weight. Now, if we have a tube of blood and we put it in the centrifugation and we separate it, we will end up having two major constituents, the liquid portion, which is plasma, and the solid portion, which is the red blood cells. So let's go deep into this. This is the tube that we have after we centrifuge it. We will have the three layers, the plasma layer, which constitute 55% of the whole blood in the tube, the buffy coat, which contains the white blood cells and the platelets, and at the bottom, we will have the red blood cells. Now, the buffy coat and red blood cells constitute 45% of the whole blood in the tube, and these are the cellular components. Now, let's talk more about plasma portion. In the plasma, there is 90% water, and the remaining 10% are proteins, carbohydrates, lipids, vitamins, hormones, and enzymes. For this reason, when we want to do a test for clinical chemistry test or immunology test or any test for hormones, we always use the tube that contain the anticoagulant. So why do we use these tubes? Plasma is the portion where there is no coagulation, because coagulation is prevented by the use of anticoagulants tube that I mentioned before. This portion contains the protein fibrinogen. These are the tubes that we use for anticoagulants: the EDTA tube, the purple tube, heparin, the green tube, and citrate, the blue tube. On the other hand, serum does not contain any fibrinogen, because the blood clot's fibrin threads were formed using fibrinogen. So this is the difference between serum and plasma. Now, what about red blood cells? Red blood cells are the most numerous cells in the blood. They are not true cells, non-nucleated cells. And why they are pigmented in red? Because they have the hemoglobin. Their function is primarily involved in tissue respiration. They transport oxygen and carbon dioxide. On the other hand, white blood cells are true cells because they are nucleated. They are white in color, and their primary role involve in the immunology reactions to protect the body from external environment. They have 5 major classes: neutrophils, lymphocytes, monocytes, eosinophils, and basophils. Lastly, we have the platelets. They are cytoplasmic fragments of giant cells in the body, which are megakaryocytes. They are also not true cells as red blood cells. They are smaller in structure. They are also referred to as thrombocytes, and their main function is to adhere to the injured area and involve in the process of sealing and healing this area. They are also involved in blood clotting, forming a clot or a thrombus. And as we know, the sources of blood are either vein puncture or skin puncture. and we will talk more when do we use the vein and when do we use the skin. Thank you for watching. Please, if you like this video, subscribe to my channel, and I will talk more about staining process in hematology in my next video.